Today's webinar will be provided by Dr. Jackie Damon, Scientific Director of Stem Cell Technologies Contract Assay Services. As a brief introduction, Contract Assay Services performs, on a fee-for-service basis, assay design and execution for hematopoietic, mesenchymal, and neural stem and progenitor cells. We offer high-content cell-based assays in a GLP-compliant environment, which allows us to perform toxicity studies on your compounds of interest. To date, we have worked with more than 80 organizations worldwide, including many of the leading pharmaceutical and biotechnology companies, resulting in the completion of over 400 studies. Our ability to perform these studies expertly and efficiently is a result of our scientific experience in studying and working with stem cells. Let me now hand you over to Dr. Damon.
Thanks, Ken, for that introduction. Hi, I'm Jackie Damon, the Director of Contract Assay Services at Stem Cell Technologies, and I've actually spent the last 20 years studying differentiation processes in erythropoiesis. In this presentation today, I will review hematopoietic stem cells and the cytokines involved in lineage commitment. I will discuss mostly red and white blood cell development and how decisions are made via binding of cytokines to their receptors, and then move on to discuss how we evaluate lineage commitment and identify factors that affect various hematopoietic lineages, both in positive and negative ways. Let's first begin then by defining what is a stem cell. The definition of a stem cell is a cell that has the ability to self-renew, which is an asymmetric division, as well as produce a daughter cell, which is functionally distinct and can differentiate into the many different cell types one can find in the body. Hematopoietic stem cells specifically are stem cells derived from adult tissue. They can be found in adult bone marrow and were first described in the 1960s by Till and McCullough. Hematopoietic stem cells are defined by three very functionally important characteristics. They can divide and differentiate, producing all the mature cell types found in the blood. They can also self-renew, maintaining the stem cell pool, and they have the ability to repopulate the entire hematopoietic system in a compromised host. The ability to in vivo repopulate is the most functional definition, is the most definitive functional characteristic of the true hematopoietic stem cell. Research over the last 40 years has taught us a lot about hematopoietic stem cells, and this has not only provided the basis of our understanding of stem cell biology, but we have also learned a lot about how to grow hematopoietic stem cells in culture and the requirements for lineage commitment to various blood cell types. This next slide outlines the hierarchy of hematopoiesis, which is a process that involves the production of billions of mature blood cells from a very small population of hematopoietic stem cells located in the bone marrow. This process, as we will discuss today, is regulated by signals from small polypeptides we call cytokines. By definition, cytokines are regulatory proteins secreted by a variety of cells. They have pleiotropic actions on cells that express their receptors, which includes activation of cell proliferation, changes in differentiation, as well as modulation of inflammatory responses. To understand how cytokines regulate hematopoiesis, we first need to review how cytokines influence and regulate the decisions cells make about proliferation and differentiation. In this next slide, we see a cartoon depicting the biochemical process required in order to transmit signals from the outside of the cell, where cytokines are circulating, to the inside, where the action takes place. Signals are transmitted within the cell by a series of protein-protein interactions known as signal transduction. This signal is initiated when the cytokine binds to its own unique receptor, a protein that spans the cell membrane. Through the receptor, the signal from the cytokine is transmitted downstream via receptor-associated proteins and a series of biochemical reactions in which proteins are modified, mostly by phosphorylation of specific amino acids. This process is controlled by the opposing activity of two kinds of enzymes within the cells: kinases, which add phosphate groups to activate proteins, and phosphatases, which remove them, turning off the signal. Kinases and phosphatases add and remove phosphate groups, thereby regulating the cell's activity and the attraction of downstream proteins through specific protein-protein interacting domains, which ultimately results in the activation of transcription factors that can bind DNA and control expression of genes involved in either proliferation and/or differentiation. Although this is not a complete list, this next slide shows the major families of cytokines and the receptors that are involved in regulating the activation as well as the growth and differentiation processes of many different types of cells that make up the functional hematopoietic system. The IL-2 family of cytokines is important in regulating immune function, for example. These cytokines work to stimulate target cells through a class of receptors that share one receptor subunit, the common gamma subunit. It is the unique alpha subunit in this family that allows the different cytokines of the IL-2 family to deliver cytokine-specific effects. Sharing receptor subunits is also seen for the GP140 family of cytokines. All the members, IL-3, IL-5, and GM-CSF, share one receptor, the beta common subunit, and specifically is again defined, and specificity is again defined by the alpha subunit in these heterodimer-type receptors. Interestingly, although they share a common subunit, the function of the cytokines in this family is diverse. IL-3 is a very important cytokine in the proliferation of the earliest hematopoietic stem and progenitor cells, for example, whereas GM-CSF and IL-5 regulate the proliferation and differentiation of later hematopoietic cells. GM-CSF acts on myeloid and dendritic cells, while IL-5 regulates eosinophil development. The next major family is based on the fact that all the receptors in this family contain a tyrosine kinase domain within the body of the receptor. This domain is important in the transmission of signals downstream of the MCSF receptor as well as c-KIT, which are important regulators of macrophage and red blood cell development respectively. EGF is also a cytokine that belongs to this group. Although it is not specific for hematopoiesis, it is an important regulator of cell growth for many different types of adult cells. Finally, the growth hormone family consists of a family of cytokines that work through specific homodimer receptors. The first two listed here, erythropoietin and thrombopoietin receptor, are critical in hematopoietic pathways and regulate the proliferation and differentiation of red blood cells and platelets, respectively. In the transmission of signals downstream of cytokines, a key component and level of regulation is the expression of the receptor on the surface of very specific cell populations. There are various families of hematopoietic receptors through which the cytokines work. Most fall into two classes with structurally similar characteristics and domains, defining them as a family we call the hematopoietic superfamily. Examples of the hematopoietic superfamily of receptors can be seen here for GM and GCSF on the right part of the slide. What is important to realize is that cytokines binding to this family of receptors require at least two receptor subunits to come together. The receptors can either be heterodimers, as we see here for GM-CSF, which consists of different receptor subunits. The shorter subunit is often referred to as the alpha subunit, and it confers cytokine specificity. The longer subunit is the beta subunit, and it is responsible for the majority of the internal cellular signaling. The other type of receptor complex is the homodimer receptors, which contain two identical subunits as seen here on the far right for GCSF. One thing the hematopoietic superfamily receptors have in common is that they are non-kinase containing receptors, which means they do not contain an intracellular kinase within their membrane, within their structure, so transmission of signals following cytokine stimulation requires downstream protein-protein interaction with the receptor and an associated kinase. The other type of receptors involved in the transmission of signals controlling hematopoietic development are those that do contain a kinase, as seen here for the MCSF receptor. As we discussed earlier, a kinase has the ability to phosphorylate and activate the cell, and when its enzyme is within the receptor itself, transmission of signals downstream involves direct interaction first of the receptor by cross-phosphorylation due to two receptor subunits coming together in response to binding of the cytokine. That way the kinase of one subunit can phosphorylate and activate the other subunit and vice versa. Transmission of the signal downstream is then carried out by downstream proteins binding directly to the receptor. For the hematopoietic superfamily of receptors that do not contain an internal kinase domain, transmission of signals downstream is carried out through a family of receptor-associated kinases as depicted in the cartoon in this next slide. These are known as the JAK family of kinases, and they can cross-phosphorylate amino acids within the receptor that cause the receptor to be modified and activated. As seen in this cartoon, JAK family kinases phosphorylate another family of downstream molecules, the signal transducers and activators of transcription, or STAT family of proteins. This family of proteins is unique in that they pass the signal directly into the nucleus. Once activated by phosphorylation by JAK, the STAT family proteins dimerize and then translocate into the nucleus of the cell, regulating expression of genes that have upstream genetic elements to which STATs have been shown to bind. This next slide shows that most receptors of the hematopoietic superfamily transmit signals through the JAK-STAT pathway, and some involve more than one member of this family in transmission of a signal within the cell. To summarize, hematopoiesis is a process controlled by cytokines which direct lineage commitment, and as we have just discussed, is regulated by the expression of receptors on the target cell, as well as intracellular biochemical signals including JAK kinases and a family of transcription factors called STATs. Let's now focus our attention on cytokines and the assays we use to study how they regulate specific lineages within hematopoietic development. As we can see in this cartoon, the earliest hematopoietic stem cell, which is mostly quiescent, is first kicked into cycle and then induced to differentiate, which requires cytokines such as IL-3, IL-6, stem cell factor, and EPO. At this early point, there is a split which results in separate lineage-specific progenitors that are responsible for lymphoid as well as myeloid development. The lymphoid cells found in the blood are dependent on cytokines like IL-7, as seen on the right part of the slide. On the other side, we see the cytokines required for proliferation and differentiation of the multipotential progenitor that can develop into the mature red blood cells, mature white blood cells, and platelets. One thing that is noticeable on this side of the cartoon is that there is redundancy, and some cytokines are important in more than one pathway. IL-3, for example, supports the proliferation of early stem cells, but is also required for the proliferation of many of the mature cells found in the blood, and it is specifically required for the development of basophils and mast cells. Other cytokines are more lineage restricted and are important in regulating cells within only one pathway. An example can be seen on the far left, where EPO is the required cytokine for proliferation and differentiation of red blood cells exclusively. Despite more than 20 years of research, it has only become clear recently that in a biochemical sense, cytokines do actually instruct choices cells make in their decisions regarding lineage commitment. Historically, it has been disputed for many years whether cell extrinsic cytokines can instruct hematopoietic stem cell lineage choice or simply allow survival of cells already lineage restricted. Using new bioimaging techniques combined with hematopoietic colony-forming cell assays that allow us to look at hematopoietic progenitor cell proliferation and differentiation in clonal cultures, Tim Schroeder's group has recently showed that MCSF and GCSF do instruct lineage choice of the common myeloid progenitor, as seen in a recent publication in Science in 2009. I will now change gears and spend a couple of minutes discussing hematopoietic colony-forming assays and how they can be used to assess lineage activity and lineage determination. If we look at this slide, which shows the hierarchy of stem and lineage-committed progenitors, we can see that although mature cells are easy to distinguish morphologically, as well as by expression of markers on their cell surface, the multi- and lineage-committed progenitors all look the same. It is not easy to identify or quantitate these cells, as they are present at a much lower frequency in bone marrow than the mature cells. These progenitors, like the hematopoietic stem cell, have very limited expression of surface markers to identify them. So to evaluate the frequency and functionality of lineage-committed progenitors downstream of the hematopoietic stem cell, we can culture these cells in semi-solid media formulations which support their growth in the presence of the appropriate cytokines, of course. While these progenitors are growing in the media, they are also differentiating, and since the media used is semi-solid, the cells grow in a cluster or colony such that the mature cell that develops from the progenitor can be identified morphologically by light microscopy. This is also known as the colony-forming cell assay. I will refer to it as the CFC assay, and it is shown on this slide as the short-term culture assay. The CFC assay can be used to evaluate lineage-committed progenitors of the erythroid lineage by the presence of CFUE and BFUE colonies, as well as the myeloid lineages by the presence of the CFUE, GM, CFU-G, and CFU-M colonies, and progenitors of the megakaryocyte lineage by the presence of CFU-MK colonies. We can also assess very early multi-lineage potential progenitors by doing a long-term culture assay, which is done in a liquid media formulation on a stromal layer of cells as a substitute for the bone marrow niche, and then after 4 to 6 weeks in culture, the resulting cells are assessed for their ability to generate lineage-committed progenitors that can be identified by the shorter-term CFC assay. To characterize the frequency of a hematopoietic stem cell, on the other hand, one needs to carry out the transplantation assay, which will identify the functional ability of hematopoietic stem cells in vivo to repopulate the entire hematopoietic system in a compromised host. To look closer at the CFC assay, this next slide shows what the colonies look like that result from progenitors growing in semi-solid media after they have been cultured for 2 weeks. During proliferation, these progenitors simultaneously differentiate, resulting in a colony of mature cells that are recognizable under the microscope. In the presence of EPO, you can see on the left of this slide 2 colonies resulting from erythroid progenitors, the CFUE, which is a late, more mature progenitor and thus a smaller colony, versus the BFUE, which is an earlier progenitor. The earlier the progenitor, the larger the colony, as early progenitors have more potential to divide and differentiate, so their colonies are larger as a consequence. If we look on the right, in the presence of myeloid cytokines, we can see colonies that result from various myeloid progenitors. On the far left, a colony of macrophages resulting from a CFU-M progenitor, and next to this, a colony that has mature granulocytes as well as macrophages, which is a CFU-GM colony that results from a progenitor that sat down in the media, divided and differentiated to produce both mature macrophages and granulocytes. This colony is large and an early progenitor that has the ability to produce both these mature myeloid cell types. If we look at the top center, we can see that in this colony we observe a central component of mature erythroid cells as well as mature myeloid cells that surround this colony. This means the progenitor that produced this colony was very early and had both erythroid and myeloid potential, and we call this a mixed or CFU-GEMM progenitor. Lastly, we can see in cultures which contain TPO, IL-3, and IL-6, we can identify CFU-MK progenitors that were able to differentiate into platelets. The importance of cytokines controlling lineage commitment of progenitors can be seen in this next diagram from a cartoon and review in Nature by John Hamilton. This shows how several of the myeloid cytokines, GM, M, and GCSF, work together to regulate pro-inflammatory signals and instruct immature progenitors to mature into the various myeloid cells like macrophages or neutrophils. This process starts at the site of inflammation with stimulation from a T-cell-derived cytokine, for example, which activates mature macrophages to produce inflammatory cytokines like IL-1 and TNF. These then activate a resident tissue cell to produce cytokines like GM-CSF, G-CSF, and M-CSF. Production of these cytokines results in the stimulation of immature myeloid progenitors to make either more neutrophils and or more macrophages depending on what is required at the different stages of the pro-inflammatory response. This slide summarizes many years of research regarding the function of these important myeloid cytokines. We now know that M-CSF, for example, promotes the survival, proliferation, differentiation, as well as activation of the macrophage lineage only. It is constitutively produced by a variety of cells, including fibroblasts and bone marrow stromal cells, and serum levels of M-CSF can be increased following activation. GCSF, on the other end, has been shown to be critical for the differentiation, proliferation, as well as activation of neutrophils, and its production requires stimulation or activation of monocytes and macrophages. GM-CSF promotes alone or with a co-stimulus the survival and activation of macrophages, neutrophils, eosinophils, as well as dendritic cell maturation, and its production also requires stimulation by a variety of cells, including macrophages, T and B lymphocytes, stromal cells, and fibroblasts. The activity of each of these cytokines plays a different role in the pro-inflammatory response, and the ability of these to direct lineage commitment is an important mechanism in regulating this entire process. An example of how we can use the CFC assay to evaluate that an immature myeloid progenitor can be instructed to differentiate into the various mature white blood cell types can be seen in this next slide. If we look at whole bone marrow cells plated in semi-solid media formulations containing only one cytokine, either GCSF, MCSF, or GM-CSF, you can see here the resulting frequency of the CFC colonies following enumeration of triplicate cultures. Across the top, we have indicated the morphological identification and size, small and large CFUG colonies, followed by total CFUGs, and then CFUGM colonies that contain both macrophages and granulocytes, and finally the colonies consisting of just mature macrophages. In the highlighted column, you can see a clear dose response in the number of resulting colonies that is completely dependent on the cytokine being tested. For example, we see dose-dependent increases in the total number of CFUG colonies where we have increasing concentrations of GCSF in the culture, and very little or no change in the number of CFUM colonies found in these same cultures. Similarly, if you look at the data at the bottom of this table, you can see a dose-dependent increase in CFUM colonies in response to MCSF and no change and very few CFUG colonies. In the middle, you can see in response to GM-CSF that you get a dose-dependent increase in both CFUGs and CFUMs, highlighting how these cytokines instruct choices the progenitor cells make as they mature and differentiate. To verify that the cells within the colony are in fact mature macrophages or granulocytes, one can pluck cells from the colony out of the methylcellulose media and subject them to a cytospin so that you can stain the cells and look for the morphological. You can look at them morphologically and identify each mature cell type. In this slide, you can see cells from a CFUG colony and a CFUM colony that have been plucked and stained. You can see that the macrophages from the CFUM colony are larger cells full of vacuoles, whereas the granulocytes from the CFUG colony are smaller cells with misshapen or lobular nuclei, as is typical for neutrophils. Since we can morphologically identify mature cells resulting from a CFC assay, we can use the CFC assay in the appropriate cytokine conditions to identify the functional activity of the cytokine with regards to its ability to stimulate proliferation versus its ability to drive differentiation of progenitors to the lineage-committed cell. This slide shows data, for example, examining the number of total erythroid colonies in increasing concentrations of EPO. As we can see that here, without EPO, there are no erythroid colonies, but when EPO is added, not only do the number of colonies increase, and increase in a dose-dependent manner, but the size of the colonies is also dependent on the concentration of EPO used, as seen in the photos above. The data shows that EPO is important not only for erythroid proliferation, but is also required for erythroid-specific differentiation. Based on the data I've shown you, you can appreciate how CFC assays then can be used to determine the in vitro activity of an unknown as well as known cytokines. This slide shows an example of using the CFC assay to look at the activity of an unknown cytokine. In this figure from a recent paper by Haishan Lin et al. In Science, the functional activity of this unknown cytokine, IL-34, was compared in a CFC assay to MCSF, also known as CSF1, as well as GCSF, for which the data here is not shown. The frequency and type of colonies observed following addition of each cytokine into the media was evaluated in a blinded manner by our group, and the concentration of test cytokine as well as the control cytokines G and MCSF was kept unknown until after colony identification. From the resulting colony number and morphology, we were able to rank from highest to lowest the concentration of each cytokine tested. We were able to show that cultures with the unknown cytokine resulted in the same number of colonies with the same morphology as that observed for MCSF. Below are the cytokine stains to confirm that the colonies identified as CFUM indeed contained mature macrophages. Along with this data, along with the CFC data suggesting that this novel cytokine acted like MCSF to stimulate growth and differentiation of monocytes and macrophage progenitors, this paper also showed that this novel cytokine, IL-34 as it is now called, binds directly to the MCSF receptor, thus identifying why the functional activity of IL-34 was identical to MCSF. I will now spend a couple of minutes talking about CFC assays and how we use those assays to determine lineage commitment in response to cytokines, and how you can evaluate stimulation as well as evaluate stimulation and synergy in the proliferation of progenitors, and looking at the increased output of mature cells. As I've shown you earlier, EPO is required for the proliferation of erythroid cells, and it also stimulates proliferation. On its own, however, EPO is a poor stimulator of proliferation of red blood cell progenitors. You can see in this figure a dose-dependent increase of erythroid progenitors in response to EPO. And if you look at the photo at the bottom, the erythroid colony with no additional cytokines, you see a very, very small BFUE. If you then add saturating levels of IL-3 in an EPO dose response CFC assay, you can get more erythroid colonies, as indicated in the graph on the right by the orange line. And as seen in the photo, second from the bottom, in the presence of optimal IL-3, the size of the colony does not really change. This suggests that IL-3 synergizes with EPO to improve the number of erythroid progenitors, but not with the ability of the erythroid progenitors to grow per se. In this next slide, you can see what happens when we added IL-6. There is no increase either in erythroid colony number or size, suggesting that IL-6 does not synergize with EPO proliferation or differentiation. In this next slide, however, we show you data in which we added saturating levels of stem cell factor, which obviously synergizes with EPO. You can see from the graph on the right that we get more erythroid colonies, and from the photos with optimal stem cell factor, you can see that the size of the erythroid colonies are substantially larger. We can also see from the photos that as the concentration of EPO decreases in the presence of stem cell factor, the size of the erythroid colonies also decreases, suggesting that stem cell factor's main activity is to stimulate maximal erythroid cell proliferation on a progenitor cell basis. When you add both stem cell factor and IL-3 with increasing concentrations of EPO, you can see a further increase in the number of colonies, but the size is similar to what we saw with just stem cell factor. What this shows is that there is a maximal size that erythroid colonies can be in a given culture, as well as a maximal number of colonies based on the actual number of progenitors plated in the original sample. More cytokines can result in increases in colony numbers and or size until this maximum is reached. In summary, this data shows you how one can use CFC assays and manipulate culture conditions to study the effects of cytokines on their abilities to affect differentiation, as well as synergize and affect the number of mature cell progenitors, the number of mature cells a progenitor is able to make. IL-3 can have different effects on different progenitors, and we show on this next slide the effects of IL-3 on the number and size of CFU-G colonies in increasing concentrations of GCSF. As you can see from both the graph on the right and the photos of CFU-G colonies in the presence and absence of saturating levels of IL-3, IL-3 synergizes to increase both the number as well as the size, or cells per colony, of the CFUG colonies. In summary, the CFC assay can be used in drug development to screen for new cytokines and cytokine mimetics, as well as to screen for new compounds or new and improved cytokines that have direct synergistic effects both on proliferation as well as differentiation of the various hematopoietic progenitors. Why this is important is that cytokines have been used for many years as therapeutics to treat diseases like anemia as well as neutropenia. EPO, for example, trade name Eprix or Procrit, has been used since the early 1990s to treat anemia resulting from renal failure. More recently, new and improved growth factors modeled after GCSF, trade name Neupogen or Filgrastim, as well as EPO, are being used to treat the complications of chemotherapy as well as to increase the maximum tolerated dose of chemotherapeutics that can be used in combination with chemotherapy. In this final section, I will quickly cover how CFC assays can be used to also evaluate lineage-specific inhibitory or toxic effects. The advantage of using the hematopoietic colony-forming assay in toxicity testing are that these biological assays are well-defined and versatile, as the assays can be designed in a variety of ways to answer different kinds of questions depending on how they are set up. The CFU-GM and the CFU-MK assays have also been validated by groups involved with ECVAM, the European Center for the Validation of Alternative Methods, whom have shown that these CFC assays are predictive of the clinical outcome of hematological diseases such as neutropenia and thrombocytopenia. Groups like Piscina's have now published how the CFU-GM assay can be used to therefore predict the maximum tolerated dose of chemotherapeutics. This next slide shows an example of how the CFC assay can be used to determine the IC50s for both CFU-M and CFU-G progenitors in response to kinase inhibitors known to inhibit kinases involved in downstream signaling pathways of these cytokines. In this experiment, we evaluated specific toxic effects on granulocytes versus monocyte macrophage progenitors following treatment of cultures with two kinase inhibitors, sunitinib and imatinib. Interestingly, we found a 25-fold difference between the IC50s for CFU-Ms versus G progenitors for sunitinib, and an even bigger difference, 500-fold, in response to imatinib. This data shows that one can determine toxicity using a CFC assay and can even evaluate lineage-specific toxicity by using the appropriate cytokine in the media formulation in which the progenitors are cultured. The ability to look at specific lineage effects between granulocytes versus macrophages is advantageous in the development of new therapeutic strategies for patients with inflammatory or autoimmune disorders. In fact, there are clinical trials now underway, some of which are depicted in this slide, aimed at targeting or blocking activity through GM-CSF and MCSF receptors as treatments for rheumatoid arthritis. The advantage of CFC assays is they can be used in the evaluation of the specificity of these drugs to target these pathways. They can be used to help distinguish the ability of targets to block GM and or MCSF activity versus effects on granulocyte progenitors to ensure there are minimal effects on GCSF targeted pathways, so neutrophil development is minimally affected. In summary, in vitro CFC assays using primary hematopoietic cells can evaluate both the stimulatory and or toxic effects of novel drugs, and there are several advantages in using these assays. The first is that these assays provide clinically relevant information, as evident by the fact that the CFU-GM and the CFU-MK assays have been validated to predict maximum tolerated dose for chemotherapeutics. These assays can be used to generate IC as well as EC50 values, allowing one to evaluate and compare inhibitory or stimulatory activity of various groups of compounds, and they use primary hematopoietic stem cells that are easily accessible as well as sensitive to drugs at various stages of blood cell development. There are also CFC assay conditions that allow for the examination of various cell lineages, and these assays can be performed using cells from various species. rat, canine, and mouse, for example, allowing one to evaluate and compare effects of compounds to identify eventually the appropriate species for subsequent evaluation of in vivo effects. CFC assays have also been shown to be robust as well as reproducible, and have even been used to do analysis of drug efficacy directly on certain disease cells. I would like to end today by thanking the people that helped provide data and put this talk together, the members of the contract assay team at Stem Cell Technologies. Lastly, I would like to leave you with a reminder that Stem Cell Technologies Contract Assay Service Department provides fee-for-service assay design and execution for hematopoietic, mesenchymal, and neural stem cells. And for more information, you can contact me directly through our website, www.contractassay.com. Thank you.